genes of multiple alleles are found in a cell and in an individual because or it is due to the presence of only two chromosomes of the same type. But in gametes contains only one chromosome, so that one allele is present, one allele of the multiple series is present in a gamete. Individuals nalle first ಏನು ಹೇಳಿದಿವಿ character nalle same locus of the homologous chromosome. Second ಏನು ಹೇಳಿದಿವಿ cell nalle ಮತ್ತು individual nalle two genes of multiple alleles are present ಅಂತ ಹೇಳಿದ್ವಿ. Third character ಏನು ಹೇಳಿದ್ವಿ gametes nalle ಒಂದೇ chromosome ಇರ್ತದೆ, ಅದಕ್ಕೆ one gene of multiple series is present in gamete ಅಂತ ಹೇಳಿದ್ವಿ. These are the three characters. Fourth character is there is no crossing over. Crossing over does not occurs in multiple alleles. You know that one, crossing over during cell division, which leads to exchange of segments of the chromosomes. That will not occurs in multiple alleles. Crossing over does not takes place. And then multiple alleles are related as dominant and recessive. Multiple alleles are related as dominant and recessive. And lastly, another character is multiple alleles controls the same character, but characterized by different manifestations. Multiple alleles controls the same character. For example, coat color in rabbit, only one character, but with different manifestations like brown, albino, Himalayan albino, chinchilla. Blood group, one character, but with different manifestations A, B, AB and O, like this. So these are the characters of multiple alleles. Sometimes or many times it has been asked for 5 marks. Define multiple alleles with an example. Give its characters. First one, located in the same locus of the homologous chromosome. Second one, in an individual or in a cell, two genes of multiple alleles are present because two chromosomes of the same type are there. But in a gamete, which contains only one gene of multiple series, then multiple alleles are related as dominant and recessive. There is no crossing over in multiple alleles. Multiple alleles controls one character but characterized by different manifestations. So these are the characters of multiple alleles. So next is a very good example that we're going to discuss now is in detailed study. ABO blood group or ABO blood groups in human beings. Multiple alleles concept was first introduced by T.H. Morgan. But ABO system of blood grouping was first introduced by Karl Landsteiner. Introduced by Karl Landsteiner in the year 1906, and he was awarded the Nobel Prize for his work in the year 1930. Okay. Before going to the study of about the blood group of human beings, let us study what is blood transfusion. What is blood transfusion? As we know, all of us know that blood will be given to the patients. That is what we call blood transfusion. So can you define the blood transfusion in biological terms? Biologically, blood transfusion is defined as injecting of a blood from a person. Injecting of a blood from a person called donor. From a person called donor to a person called recipient, to make good to the loss of blood in recipient's body. ಒಂದು ಬಾಡಿಯಿಂದ ಮತ್ತೊಂದು ಬಾಡಿಗೆ ಬ್ಲಡ್ ಅನ್ನು ಇನ್ಜೆಕ್ಟ್ ಮಾಡೋದ್ರಿ. ಬ್ಲಡ್ ಅನ್ನು ಕುಡಿಯಕ್ಕೆ ಬರಂಗಿಲ್ಲ, ಬ್ಲಡ್ ಅನ್ನು ಎಲ್ಲಿ ಬೇಕಲ್ಲಿ ಹಾಕಕ್ಕೆ ಬರಂಗಿಲ್ಲ. ಡೈರೆಕ್ಟ್ಲಿ ವಿ ಹ್ಯಾವ್ ಟು ಇನ್ಜೆಕ್ಟ್ ದ ಬ್ಲಡ್. Injecting of a blood from a person called donor to the person called recipient. This is called blood transfusion. So blood transfusion, it is there since from olden days, since from ancient days. And we know that our Ayurvedic medicines or Ayurvedic surgeons will do it, will done that one in ancient time also. But sometimes that will be matched with the recipient's body, so the patient becomes normal, and sometimes it will be mismatched in the recipient's body, leads to death of the recipient or leads to death of the patient on the table, operation table itself. Why it is so? So the transfusion of blood can be done during surgery or during anemic conditions. Anemic condition means during loss of blood. At that time, so blood from an individual cannot be safely mixed with the blood of another without leading to a disastrous condition, without leading to a disastrous result. It cannot be safely mixed. It is mainly due to agglutination or due to clumping, which may or may not occur after transfusion. ಮೊದಲೇಕ್ ನಮಗೆ ಗೊತ್ತಿದ್ದಿಲ್ಲ, ಬ್ಲಡ್ ರೆಡ್ ಐತಿ, ಲಿಕ್ವಿಡ್ ಐತಿ, ಒಂದೇ ಥರ ಐತಿ. ಸೊ ಬ್ಲಡ್ಡನ್ನು ಹಾಗೆ ಹಾಕಿಬಿಡ್ತಿದ್ವಿ, ಇಂಜೆಕ್ಟ್ ಮಾಡಿಬಿಡ್ತಿದ್ರು. ಕೆಲವೊಂದಿಷ್ಟು ಟೈಮ್ ಏನಾಗ್ತಾ ಇತ್ತು, it results in a dangerous condition, resulting to the death of that patient, which may cause death of that patient because of agglutination, because of clumping, resulting into that condition, resulting into that status. So which cannot be. So extensive studies have revealed that the agglutination or the clumping is due to the reaction between two substances present in the body. Two substances present in the body, that is in the blood. One is antigen, another one is antibody. Agglutination or clumping agglutination or clumping is mainly due to the reaction between antigen and antibodies. Due to the reaction between antigen and antibodies. So what are these antigens and antibodies now? Antigen or agglutinogen. Antigen or agglutinogen